at this time. And it's just a venue to learn about and talk about new, or not necessarily even new, just tools, technologies, methodologies, and various stages of development. Some maybe are just in development, others maybe have been around for a while but are used in some novel way. And so just to have an opportunity to talk about these tools. There is a sign-in sheet is hopefully floating around. Please do sign in. It helps with the pizza to ensure that we have enough pizza for future weeks. And with that, I'm going to actually let Maureen introduce our speaker.
Thanks, Marcy. So I'm happy to introduce my student, Raymond Cavalcante. He's been in my lab since, 2012? Yeah. So he is a senior graduate student finishing up. He has 2 first author publications so far in my lab. And this is part of his 3rd project. We started developing Annotator. Actually, it was Raymond's idea because we saw a gap and a need. A fairly simple conceptual idea, but there was a gap that needed to be filled, we thought. And so that's what he's going to be talking about today. I'll turn it over to you, Raymond.
Okay. Thank you all for coming. So I was going to talk about a tool that I've been developing and that is pretty much developed.
Check to the person in the back.
Yeah. I'm going to tell you about a tool that I've been developing called Annotator. And the idea is it's going to help you interpret your genomic regions that you might have. So kind of why would anybody want to do this? The idea is we have a lot of data, right? If you do ChIP-seq, then you have a bunch of regions that represent transcription factor binding. If you do bisulfite or RBS sequencing, you might have a bunch of CpG sites that have methylation associated with them. If you do MEDIP or HMEDIP, you have regions that are associated with methylation. Or if you do DNase or ATAC-seq, then you might have a bunch of regions with associated open chromatin. And you don't want to just look at a table that gives you the locations of them. You want to know where in the genome are they and what features do they intersect with. And so you want to know, does that transcription factor mostly bind to promoters? Is that SNP in an exon? Is the methylation lower in CpG islands than it is compared to other regions? Is there an enhancer that has open chromatin around it also, right? So you want to kind of answer these questions to help interpret your data and maybe generate some hypotheses along the way. So that's kind of why anybody might want to do this. And so kind of as Maureen said, the reason we want to do this is because we're developing a DNA methylation analysis pipeline that we call MINT. And we have a bunch of data associated with that pipeline, like CpGs with percent methylation and coverage information, or differentially methylated regions with hyper or hypo methylated labels. And then we also have a classifier that gives us a bunch of categorical labels for the regions. And so we want to be able to interpret that data besides just staring at a large table of regions and labels. And kind of as a side thing, last fall when I started developing this with Hani Habra, we were both taking Biostat 615, so I needed a project. And this was kind of a perfect little thing that took up a whole semester. So there are a variety of existing tools that kind of deal with this problem, but we found that we didn't really like any of them. So one that we found early on, and that seems to be used by a lot of people, is ChIP-seq Anno, which is an R package. And so one of the problems that we had with it was it's pretty slow, and I'll get to that later. Another problem is that it imposes a prioritization on the annotations. So for instance, if a region is in a promoter and a CpG island, it chooses for you. Or you could choose for yourself, but nonetheless, you're only restricted to one annotation per region. And that's not realistic, and you might want to know that it's in both of them. And we found that their visualization functions weren't particularly helpful for what we wanted to look at. So this is an example of the pie chart that tells you kind of how the region falls inside of features. But we wanted more information than that. Another option is Homer. And while I really enjoy their logo, and it is faster than ChIP-seq Anno, it also has the problem of imposing the prioritization, which we're not into. And it doesn't do visualizations. We would have had to do it ourselves anyway. So that's not really good. And so the first thing that I implemented was, for MINT, was using bed tools, which is a really versatile and wonderful application, and it has lots of uses. So you might want to look into it. I implemented something with bed tools in ggplot2, and it was kind of hacky, and I had to go get the annotations myself and parse them, and then I had to build the visualizations myself. And I thought, why don't I just generalize this and do everything in R? and make a nice R package. This gives me a chance to build an R package from scratch, which I've never done, and so it lets me play by my rules, which I kind of wanted to mess around with. So there are some goals for Annotator. We need to be able to read a variety of file types quickly, right? Like you could have bed files, or you could have whatever weird files, Bismarck outputs that aren't bed files. Or you might have something altogether different. So we have to be flexible in terms of the type and the size of the file that we want to read in. We also want to annotate to common and custom annotation types. So we have a bunch of GENIC and CPG features for human and mouse genomes to start that are built into the package. And we also have some custom annotation capabilities. And we also, like I mentioned a couple of times, we want to return all of the intersections, not just a prioritization, so that you can have kind of the full context of where your regions are falling in the genome. And then we want to summarize and plot over any of the categorical and numerical data that might be associated with your regions. So the implementation is exactly along those lines, right? So it's divided into modules that read files or data frames if you're already in an R session, annotates the regions to genomic regions, and then summarizes and plots them along with their data. And so from here on out, I'm going to illustrate how Annotator works by walking through kind of an example with particular data. So I don't want to dwell too much on the biology of this example data, but I just want to point out that it's differential hydroxymethylation data between two conditions. Or sorry, not hydroxy, just regular methylation, using a tool that was developed in our lab called MethylSig. And so this data is particularly nice for illustrating Annotator's capabilities because it has a categorical data variable, it has a bunch of numerical data variables that we can kind of play with all of our visualization and summarization options. And of course, there are regions, so that's great. So
it went very fast, but looks like the first. Oh no, I take it back.
Okay. So I guess it is worth mentioning some of these things. So for example, we're going to talk about the differential methylation status, which can be none, hyper, or hypo. And then we have numerical values like the difference in methylation between group 1 and group 2, and then we have the individual group methylations in those regions, right? So we're going to play with that data in the visualizations. So kind of the first thing that we want to do is we want to be able to read small files, like maybe tens of thousands of ChIP-seq peaks that come in like narrow peak or broad peak if you get them straight from ENCODE. Or we want to be able to read large files that have like millions of CPG sites like we're encountering with our Mint pipeline. And we want to be able to do that quickly. And so instead of using the base R package, I'm using Reader, which was developed by Hadley Wickham, and it's kind of an alternative to like data.table and the fread or whatever that function is called. So the Reader package allows us to quickly and flexibly read any kind of file type we want in, and then we're going to store it as genomic ranges because that's a convenient data type, and there's lots of optimized methods for it. So this is I'll kind of give you code snippets for how like your analysis would be coded to kind of demonstrate that it's not a lot of coding, and it's quite simple. So this is the function that reads the regions for the data that I showed you just in the previous slides. So you can specify what the column names are, and you're probably going to want to do that because you'll refer to them later in the visualization and summarization functions. And you can specify what kind of column types you want. Specifically, you can kind of decide, I don't want to read in these columns because I want to save myself time and space. So that's what allows you to read in kind of any type of file that you like. So as an example, right, if we have some random max 2 peaks, narrow peak, we're obviously interested in the locations of the regions. We might care about the names, and then we want to pull out the fold change and the p-values, right? And so we name the columns accordingly. And then we tell the read function which ones to skip by using a dash. It's just kind of, I've inherited the syntax from the Reader package. So this kind of allows us to read in, like, I don't know, like 25 million regions pretty quickly without having to, yeah. And so one of the other things is I didn't want people to have to mess with their files beforehand to conform to something that we wanted them to have, because I didn't want to do that for everything in our pipeline. So I figured this was kind of the best split the difference option. OK, so after you read in all your data, then you want to annotate it, obviously. So I'll go through kind of what annotations we have. So there's a companion package called Annotator Data, and it has human and mouse annotations for CPG-related features, and also for genic annotations, and included are Entree gene ID and symbols, so you can easily search the results for that. And then we have an enhancer annotation for HG19, and we want to add some more for mouse and maybe different annotations for enhancers because they're quite variable, as you may well know. So our CPG annotation is quite simple. We just grab the CPG islands from known genes, and then we kind of build out into KB chunks. And we make sure that none of the islands intersect the shores, and none of the shores intersect the islands, and so on and so forth. So this is a partition of the genome. So that's pretty simple. But our genic annotations can get kind of complicated, or you can keep it basic if you like, right? You can have promoters, 5' UTRs, introns and exons, and then the 3' UTR, or you can split up, the introns and exons, whether they occur in the 5' UTR, the 3' UTR, or the CDS. If you're into that, you don't have to be. And then we have the 1 to 5 KB upstream. It means you can kind of increase the resolution of whether your regions intersect like an exon that's in a 5' UTR or just in a CDS, or that kind of thing, if it's of interest to you. So not pictured in this kind of complicated graphic is we included intron-exon boundaries, and those are defined as plus or minus 200 base pairs between or around the junctions. And then we have the intergenic regions, of course, which are where there are no genic annotations. So like I mentioned before, we also support custom annotations. So the function that does that is very similar to the read function, except you can read in any number of custom annotations at the same time. You just have to specify where they are and how they're structured in lists, right? So that's pretty basic. So in this case, these will carry on through to the figures and the summarizations that we see later. So I've just grabbed some EZH2 peaks from GM12878 and some CGN peaks from K562 from ENCODE, just to kind of give a flavor for what's possible. You could include DNase peaks, whatever you want. So in order to annotate the regions, like I mentioned before, we're using genomic ranges because it's really highly optimized and it's very fast. And then we use some dplyr stuff to kind of collate all the results together. So in order to do this, really all you need to say is what annotations do you want to annotate to, right? And so instead of having to type out like CPG islands, CPG shores, CPG shelves, etc., I made some little handy shortcuts, right? So you don't have to do all that typing. So that's just a cute little feature. So you give the list of what annotations you want. You say what the regions are, what the annotations are, and whether or not you want to ignore the strand. In this case, we're going to ignore the strand. So that's all it takes to annotate the regions. So this is kind of the big table that maybe you don't want to look at so much, and why we have the summarization and plot features. So this is actually 4 lines, but it wraps. So the whole point is the result of the annotation gives you information about the annotations, like the annotation type, where the annotations are, and maybe the Entree gene ID and symbol in case you want to search on that table. And then all the information that comes with the data, right? So where the data is and all of its corresponding numerical and categorical stuff. So you may want to interact with this table, you may not. It's up to you. So I'll start talking about now the functions that kind of help you interpret the data beyond the annotation table. So the summarize functions basically just give you more tables, but they're smaller. So a very obvious thing you might want to do is just how many regions do I have per annotation, right? So that's like a table function, basically, but I figured, well, why not just include it in the package? So all you have to do is give it the table of the annotations, and then it will give you the annotation types and how many there are. It's very basic. But you might be interested to know, like, how does that compare to like a background set of regions, or like a random set of regions, or something like that, to figure out if like this 5,653 is like special to the data or not. So we included some function, a function, to let you create kind of a randomized set of regions based on your input data. So it's length and chromosome matched if you want chromosome matched. You probably do. So in order to do that, you just need to give it your regions, what genome you have, so you're not like jumping outside of the genome bounds. And then this creates kind of a set of random regions, and then you just need to annotate them again. So whereas in this function, we just gave it the annotated regions to figure out what that table looks like with the random regions, you just give it both of them, right? And so you'll get a longer table. It'll have an extra column that tells you information about how many there are in the data and then how many there are in the random regions. So then you can kind of make an assessment about if that's abnormal or not. Okay, so something else you might want to do is take a numerical column and look at how that value looks over the annotations for all of the regions that intersect that annotation. So you can use the summarize numerical function by giving it the annotated regions, of course, and then telling it what categorical columns you want to group the regions by. So in this case, I want to group them by the annotation type and the annotation ID. And then I want to do the summary over the difference in methylation. So what does this give us? This tells us for each unique annotation type and ID, how many regions intersect it, and then what is the mean of that value? What is the standard deviation of that value? So it's just a way to look at your data like with respect to any grouping of categorical data that you want. I found this to be kind of the more obviously useful one. And then something else you might want to do.
Why do you use so many significant figures over here for the mean and the standard deviation?
Say again?
Why do you use so many decimal places?
Oh, that was just the output. I didn't.
Is it easy to, you're writing code all the time.
Yeah.
To cut it off at 2 decimal places?
Yeah, I could do that.
Be more
yeah.
justifiable.
Right. So the next kind of thing you might want to do is just count how your regions fall by grouping among any kind of other categorical data, right? So you don't have numerical data necessarily. So in this case, I was interested in how does the differential methylation status, how many regions of each differential methylation status type are in each annotation type, right? So split the annotations by a certain categorical variable. So this is kind of what that gives you. So you can do numerical summarization or categorical summarization. It's kind of those two functions. So I've also included a bunch of plot functions. And so for these ones, since the code doesn't fit with the plot on the same slide, I'll kind of show you the code and then show you the plot. So again, you might want to know how many regions are there per annotation type. And so there's a function called plot annotation that gives you a bar graph, which we'll see in the next slide. So basically, you need to give it your annotated regions. What order do you want the annotations to occur in? You don't have to give it this, but you can if you want. And this vector also serves as a subset function. So you don't have to give it all of them. You can give it some of them. And so when you do that, you get just a regular old bar graph that looks just like what that table would look like if you made it a bar graph. And again, if you wanted to look at what it looked like with random regions, which I would argue is probably more interesting, you just need to add the random regions, right? And so then you get a dodged bar plot that maybe in this example tells you, oh, there's a lot more regions in my data that are in enhancers than I would expect from the background. Or maybe there's less intergenic in my data than I would expect versus background. Very basic stuff. So another kind of thing you can do is you can do plots of numerical data, a single numerical data column, over kind of any facet. So I've kind of imported the ggplot2 language here a little bit. So you can tell it to show you all of the methylation, the distribution of the methylation in group 0 over all of the different annotation types, right? And so this is what that looks like. And so we have the group 0 methylation rates in each of the regions that fall into the different annotations, right? And so the gray bars are the distribution for the regions that are in that particular facet, whereas the outlined red bars are kind of the overall distribution. So in this example, you can see that promoters look like they're hypomethylated, whereas maybe the intergenic or the 3' UTRs seem to be a little more methylated than background. So depending on what your data looks like, this might be more interesting or less interesting. I don't know. So you can also add another numerical column to the very same function, x and y, and you can just have a scatter plot that's basically the same thing, right? So in this case, I'm comparing group 0 methylation versus group 1 methylation over the different annotation types. So you could also do like a volcano plot or something like that.
So can I send like the gene list? Like, can I give my own GTF file here?
For like the annotations?
Yeah, for HC19, if I want to select like particular gene list, can I give that here?
You could use it as a custom annotation, yeah.
So Raymond, if he did that, he would have to specify out all of the promoters and 5' UTRs.
Yeah, it depends on how detailed you want it. If you just want like the gene body, then you could just use the start, stop, and the GTF file. But if you want like the exons and stuff, you'll have to.
So if I select few genes from the GTF, so I need to give all this information, like promoter of all this information, or I just what information I need to give here actually?
Oh, so there's on the previous slide where I showed you how to read in the custom annotations. You could just give the GTF file and specify which columns you wanted to grab out of it. Right? And then that would be like one annotation. You would subset it beforehand, and then that would be like one of the facets, like your custom genes or whatever. Yeah, that's a good point. Okay, so you can kind of plot numerical data over a single column or over 2 columns if you like, depending on what kind of data you have. So you might also be interested to know how are regions co-annotated? And so I've created a function called plot co-annotations that looks for exactly that and gives you a heat map. So the function is quite simple. You give it your annotated regions like all the other ones and what order you want them to be in. And you get a heat map that tells you, okay, well, for example, I have maybe 163 intronic enhancers, or I have 1900 CPG islands that fall in a promoter, right? So you might be interested in particularly those regions. So you can, based on this and the numbers that you get, you can go subset that table, or if you're interested, you can plot numerical data over those co-annotations. So you could say, I want to look at the methylation rate in group 0 over the annotations of CPG islands and promoters. And what you'll get is a multifaceted histogram that tells you where the gray bar, in this case, this is all of the regions that are just CPG islands, all of the regions that are just promoters, and then all of the regions that are both CPG islands and promoters. So you can kind of see the difference between whether it's important that it's in both or not. Right, so in this case, you see that maybe there's some more hypomethylation in this group when it's both annotations, as opposed to either one individually. Okay, so that's kind of all of the numerical data stuff. So then there's just a couple more functions to do categorical things. So you might be interested in splitting your regions by whether they're hyper or hypomethylated, and then within both of those, you might want to know how are the annotations distributed. So this is a categorical plot, and where you tell it, okay, I want to split it up by the differential methylation status on the x-axis, and then I want to fill those bars with the annotation type. And so this is exactly what you get, right? So this is the distribution of all of the sites that I tested for differential methylation, how they fall into the annotations. And then this is the number of hypermethylated regions and hypomethylated regions also split out among the annotations. But obviously those are a little squished and you can't really figure out what's going on there necessarily other than to say, oh, it looks like there's a lot of CPG islands. So you might want to do the same thing except replace this position parameter with fill so that it gives you the proportions instead. And so you can kind of see immediately that in the hypermethylated, there's a lot more regions occurring in the CPG islands and shores versus kind of what I would say the background is, or even versus the hypo, whereas hypomethylation is occurring more in intergenic regions, or inter-CGI regions. Right, so this gives you an idea of kind of how your data is behaving. And this function is flexible in that you can give it, you can put any categorical columns in the X or the fill, right? So I could flip the differential methylation status and the annotation type and end up with a plot that tells me what is the relative hyper and hypomethylation in each of the annotations. And maybe you like looking at it this way better than the other way, and it's up to you. So you can kind of see that it looks like there's a lot more hypermethylation in enhancers in this relative to all of the regions and versus kind of some of the others, and so on and so forth. So those are kind of all of the summarization, plotting, annotation, all that kind of stuff. That's kind of the meat of the package. And so I went ahead and benchmarked this against Chipicano. So when I said Chipicano was slow, I meant this slow. So we compared Chipicano and Annotator from read-in to annotation because they don't do visualization, so it's not fair. And we used known gene annotations for both, and then I gave it 4 different file sizes, right? Small, 27,000, 365,000, all the way up to 25 million, right? And so bluish is Chipicano and red is Annotator, and you can see that for small ones, it's like, well, what's 2 seconds? But as you get larger, the difference is quite stark. So I'm hoping that this makes people want to use Annotator over something like Chipicano. So I hope I've convinced you that Annotator is pretty fast, that it's rather flexible, and that it's relatively easy to use with all the code snippets. So if you happen to have genomic regions and you want to annotate them, I'd really be happy if you used it and you gave me some feedback. And if you don't like features or you want to see certain features, I'd more than happy to hear all about that. So as far as where to get it, right now it's on GitHub, the Annotator data package and the Annotator package both. And you can install them using this simple command. And Maureen was kind enough to let me submit this to the bioarchive. So if you don't know what the bioarchive is, it's like a preprint server for bioinformatics and other biology-related manuscripts, kind of like the physics archive, right? So we put this on the bioarchive maybe in January. I don't know, it was a while ago. Yeah. And we're going to submit it to bioinformatics as an application note soon, hopefully. And I've submitted it to Bioconductor, and I'm kind of in the middle of negotiating what I want to change and what they want me to change. So we'll see how long that takes. So with that, I'm happy to take any of your questions.
So Raymond, some people here might be interested to know like how submitting a package to Bioconductor has evolved and what you're going through now.
Oh, yeah, so they've gotten a lot more stringent. So you basically submit your repository over GitHub to their as an issue, and then they kind of look at it for like scientific content to make sure that it's not replicating other things, and then they kind of, if you re-implement anything that they've done in a package, they really don't want you to do that anymore. And so they try to get you to use their infrastructure, which sometimes is okay and sometimes isn't. So yeah, they're paying a lot more attention to the quality of your code and duplicating functionality and all of that kind of stuff, which is good, but yeah, a little frustrating sometimes.
Well, it wasn't just that, but they were asking Raymond to add something to one of their packages.
Right.
We could use it from there.
Yeah, they were complaining.
That was going a little too far.
Yeah, they had complained that I made my own annotations when they have their annotation database, but like they don't have CPG islands for HG38. So what am I gonna do? Yeah. Anyway,
yeah,
that was
so
a comment.
Raymond seems like a great sort of work with like improve performance and everything, and I like visualization. So the question is, I assume that you documented all those things that in the package that you talked about here.
Yeah, so that was one.
I guess I mean, obviously performance is one thing, but also the ease of use. I mean, it's all obvious to you, and
right.
as you were
I've been
describing
staring
it,
at it forever.
It's sort of obvious to me, but I don't think it will be if I go and try to do it on my own. So.
Right. Yeah. So I have a pretty detailed vignette.
Okay,
that kind of step. This is basically the vignette, actually.
Okay.
And then all of the manual pages are pretty filled out with examples and all of that kind of stuff. I tried to be as thorough as possible.
Great.
Thanks.
So when you depart head on to the next stage of your career, what kind of maintenance effort is required to keep this useful and current?
So yeah. So if we're going to continue to do our own annotations, then I would have to kind of rebuild the annotations for new genome versions as they become available, right? So that's one of the advantages of Bioconductor telling me use our packages, because then it's easier easier to update with any new genome just by calling their stuff. So yeah, I guess code base wise it's not too tough, but it's the annotations that are what's really important. And I guess it's keeping up with whatever package dependency changes occur. I've already encountered some visualizations changing because of the underlying dependencies. So it's not trivial, I guess. Yeah.
That was really nice and excited to use it. My question, I guess, is I like that you're being inclusive. So, every annotation doesn't override other annotations, but I'm wondering if you put any thought into doing the reverse, where you're sort of what I call more doing roll-ups in terms of if I want to annotate by gene and I don't want to have 10 pieces of information associated with that gene, I want to sort of collapse things rather than expand.
Yeah.
Have you thought about how to do that?
I guess one quick thing would be to just include an annotation that has the gene boundaries, right? So it's just the gene body itself from TSS to TES, and that could
just isolate it that way.
Right.
Okay.
Yeah.
That doesn't summarize the the data associated with it. I guess that's the problem, right? Like if you have readings in your methylation, they're sort of up and down within that area.
Yeah, I guess you would.
What I'm getting.
Yeah, you could use the summarize function, and it would kind of figure out what the different methylation is over all of the regions intersecting that gene body, but you would, I guess, not get to see the distribution in the gene body.
So you just take the average? Is that the...
Yeah, that's just kind of the first pass.
Okay.
It could be more complicated, always.
So I like that, excuse me, I like that you include the ability to pull out a randomized background set of regions. And my question is, so do you have any functionality in there to perform like statistical tests for enrichment against that background, or even as simple as putting like error bars on your histograms or anything like that?
No, we didn't include any tests because we were kind of nervous that the same test won't be applicable in all cases. So we didn't want to create this one-size-fits-all thing. So I figured it's easier for me to just not do it. So it's left to the user as an exercise.
Yeah, in fact, we talked about this just this
couple days ago.
Yeah, because I feel like when you develop a tool, you want to be careful to create it so that it's not easy for the user to screw up, basically. It's not easy for them to get a biased result in a statistical test.
Yeah.
And yeah, we haven't been able to figure out a way to be pretty sure that they won't do that.
Okay.
Yeah.
It's kind of tricky.
Second question related to the background is, why do you just look at chromosome and length rather than like including things like GC content or distance to a transcription start site?
So under the hood, we're using the regioner package, and you can get more fancy. But kind of as the first implementation, I just did the length and chromosome matching. And we could conceivably add other parameters for like qualities that you want in your regions, but yeah, I recognize that's something people would want.
Is that something that you do?
Yes.
Yeah.
Okay. What other parameters do you use?
Yeah, what else?
So mostly just GC and length to distance to a TSS, because I'm looking at regulatory elements. So
okay.
Those are particularly important variables to what I do. So.
Yeah.
So my question is about, you actually sometimes have many-to-many mapping. Not only one region, multiple annotations. Sometimes one annotation is broad enough that it's multiple of the regions.
Right.
If you do have conflicting data points, like 3 hyper and 2 hyper, how do people generally summarize?
So you mean there could be
if a gene body is long, I get 3 hyper methylated 2 hypo, or I have this island surrounded by shore and ocean. I might get something that's not easily summarized, discordant data. But as my question is more general, I wonder about it.
Yeah, I guess it depends on the context as to what you would do. But Raymond could say specifically what the plots do in that case.
I know yours is visualization annotation, but algorithm-wise, how do people in the field boil it down to like 1 score per gene, 1 score per enhancer?
Yeah.
Yeah, so it's tricky when you're comparing across regions like that. And there's one specific paper. Do you remember the?
Oh, Leo Proctor and his student about the Simpsons paradox. Is that what you were talking about?
Yeah, we can talk. I can talk to you about that afterwards if you want. But yeah, that often happens that in a region there's both hyper and hypo in slightly different regions within a region.
Right.
So, Marcy, we have for years been building up valuable tools. And in the old NCIBI website, there's a listing. In the DCMB, CCMB website, there's a carry forward listing. It might be a good session in this series sometime to actually put up the list and ask the people most knowledgeable, or maybe not even most knowledgeable, but people who've used the tools, if anyone has recently, to describe the pluses and minuses and what kinds of upgrades might now be needed to keep them useful for ourselves and for others. In general, my impression is that in bioinformatics and many other fields, people who develop new methods, new tools, tend to use their own. Occasionally, they compare with others, as Maureen has demonstrated very nicely in her old bioinformatics paper on concept gen. But the performance across our colleagues all around the community is not great for learning to use and appropriately using multiple tools. But in general, if there are 2 or 4 alternative tools, if they're current, some have this advantage and that limitation, a different advantage and different limitation, and being aware of when they might be useful, when it actually might make a biological finding more robust to show that you get the same key result for 2 or more different tools, given their differences. So that might make an interesting session, or more than one session. And I think bringing everybody in this room and your colleagues throughout the program up to speed on what we have done and what we have available. It's all available to you, and in most cases there's somebody still here who could help you use whatever sounds potentially valuable.
It's a great idea. I'll try to put something together.
Great.
Okay, well, thank you all for coming.
Thank you.